So it's a pleasure to introduce Sarah Kinkel to you today. Many of you will know Sarah. She's been working at WeHi for a while now, and she's worked in many fields related to epigenetic control. She started off working in spermatogenesis and thinking about the role of DNMT3L, which is a DNMT regulator protein, and the role in spermatogenesis, and then worked on this protein also in immune cell function, the role of medullary thymic epithelial cells, and more recently has been looking at chromatin modifiers, more specifically the polycomb repressive complex in hematopoietic stem cells. So this gives her a really strong and broad base of knowledge in epigenetic control, and she's going to relate this to disease today in her talk. So welcome, Sarah, and we look forward to your talk.
So hi, my name's Sarah. I'm actually a postdoc in Marnie's lab in the molecular medicine division, and the title of the talk I was giving today is on chromatin and disease. But what I'd really like to focus on today in particular is DNA methylation, and that is because DNA methylation is perhaps the most well-studied and probably oldest known epigenetic modification in DNA. And I'm going to use this as sort of an example of epigenetic modifications and how epigenetic coordinated gene expression helps control cell identity and function during development, and also how mistakes in this process can cause disease. So just to give you a quick seminar outline, I'll give a brief introduction to DNA methylation, about what we know about where it is in the genome and how DNA methylation patterns are established during development. And then I'll also talk about some specific diseases, some chromatin diseases, and these will include specific defects in the methylation writing and reading machinery, imprinting and imprinting disorders, and also talk about methylation of transposable elements and repeat instability diseases. So just to get started, what is DNA methylation? It's the addition of this methyl group to the 5th position of cytosine, and in mammals this occurs predominantly at Cpg dinucleotides. So when people talk about methylation, they often talk about how it is really important in some key developmental processes. And I've listed these here. So we know that DNA methylation is very important in parental imprinting. And parental imprinting is the monoallelic expression of a particular gene based on its parent of origin, so whether it's come from the sperm or the oocyte. There's also X-Chromosome Inactivation. So this is a method of dosage compensation in mammals where XX females actually silence, completely inactivate one of their X chromosomes in order to achieve dosage compensation with XY males. Also, DNA methylation is very important in the suppression of endogenous retroviruses, and this is because otherwise, if they're expressed, they can jump around and cause havoc and mutations in the genome. And also, DNA methylation is important for genomic stability. So I thought I'd give a bit of a historical overview on the study of DNA methylation and how it was determined that it's actually important for these key developmental processes. And given the previous talks that Catherine and Ian have given, I thought I couldn't really give a teaching seminar unless I included some black and white pictures of some serious-looking scientists doing some serious science. And so this guy has some particularly serious facial hair as well, I'd like to point out. But this is Roland D. Hodgkiss, and in 1948 he was actually using chromatography to isolate nucleotides from calf thymus, and he identified a 5th nucleotide which he actually called epicytosine, and this is what we know as methylcytosine. And just to place it in context of what was going on at the time, it was a few years later that the far less serious-looking Watson and Crick actually published their paper on the double helix molecular structure of DNA. And it was in the 70s that some scientists, in particular Robin Holliday and Art Riggs, first proposed the idea that perhaps DNA methylation was actually involved in controlling gene expression and gene regulation during development, and was important in giving cells their identity. And it was also in the 1970s that Nobel laureate Hamilton Smith discovered restriction enzymes. And in particular, they discovered methylation-sensitive restriction enzymes. So these enzymes can cut a specific sequence of DNA, but may not be able to cut it if, in fact, the cytosines in that sequence are methylated. And this gave researchers the first opportunity to correlate the level of methylation of a gene and its expression. And there was a paper published looking at chicken beta globin, where they found that in adult erythrocytes, the beta globin was actually unmethylated and expressed, whereas in the oviduct in the brain, you saw lack of methylation at beta globin. the gain of methylation of beta-globin, and therefore it wasn't expressed. So methylation and silencing, not methylation, expression of genes. I decided not to show any pictures of scientists in the 80s because their hairdos were way too bad. But what I have done is pointed out that this was the period of time when bisulfite sequencing was actually developed. And so bisulfite is a chemical that treats cytosine and 5-methylcytosine differently. So it allows you to PCR and sequence and actually identify which cytosines were methylated. It also saw the discovery of the DNA methyltransferase enzymes and also the identification of the first imprinted genes in mammals. And these days we have a wide range of methods that we can actually use to make genome-wide maps of DNA methylation in different cell types. These include immunoprecipitation methods, MBDFC and MEDIP, in which case we can actually enrich for methylated cytosine. And then we can combine this with whole genome sequencing or bisulfite sequencing, whole genome sequencing. And this allows us to give genome-scale DNA methylation maps. And this has been done in whole chromosomes in humans, as well as pluripotent cells, germ cells, differentiated cells, and of course, people have looked at tumour cells as well. And so I'm showing you an example here of human chromosome 22. You can see there's a scale where blue is hypermethylated and yellow is hypomethylated. And you can see along this region, there are various regions of dense CpG and different cell types shown here. And you can see the differential methylation, so very methylated, unmethylated, between different cell types for the same genes. And of course, there are some regions that are consistently methylated in all cell types analysed. So what have these genome-wide studies actually told us about the distribution of DNA methylation in the human genome? Well, there are roughly 28 million CpG dinucleotides in the human genome, and this is actually, in general, mammalian genomes are globally depleted for CpGs. Depending on the cell type you look at, they find that about 60 to 80% of CpGs are actually methylated. And the methylation patterns, they've actually done studies looking at germ cells, early embryos, and then tissues at later states. And they've shown that DNA methylation patterns are actually erased during development, particularly in the early zygote, and then re-established in a cell type specific manner during development. One of the main features we talk about when we're talking about DNA methylation is Cpg Islands, or CGIs. And these are CpG dense regions that are often enriched at the promoter regions of about 60% of human genes. And generally, these tend to remain unmethylated in different cell types. And this is particularly the case when we look at housekeeping genes, where we require the expression of these genes in every different cell type, and they tend to have unmethylated Cpg islands. But of course, there are examples of differential methylation at CGIs, and this includes on the inactive X chromosome, where of course on the inactive X you see methylation of the CGI, but not on the active X. And there are also examples of tissue-specific differential methylation, although in general, these tend to be on what they call weak or low-density Cpg islands. And these tend to be not necessarily at promoters, but often at distal enhancer elements. And these seem to dictate epigenetic tissue-specific regulation of certain genes. And of course, it's well known that DNA methylation patterns are actually disrupted in cancer. And in general, there is a global hypermethylation of DNA, which perhaps is involved in genomic stability and epigenomic instability. And there's also a specific hypermethylation of tumour suppressor genes. So in certain tumour types, you have a hypermethylation and therefore silencing of these tumour suppressors. So the enzymes that are responsible for actually laying down the DNA methylation pattern and then maintaining it through cell replication are the DNA methyltransferases, or the DNMTs. And there are three active DNMTs. DNMT1 is the maintenance methyltransferase, whereas in the de novo family of methyltransferases, DNMT3, there is DNMT3A and 3B. And they all share this conserved catalytic domain that allows DNA methylation. And then the runt of the litter is DNMT3L, which actually lacks amino acids in its catalytic domain, so it has no actual functional, catalytic activity, but it's known to bind to the other members of the DNMT3 family and stimulate their activity, and is actually quite important as well. So just to give you some information on DNMT1, this is the maintenance methyltransferase, and it's called that because of its preference for methylating hemimethylated DNA. So this is when you've got two strands of DNA and only one of those strands has methyl marks on the CpGs. And it's able to bind to this and actually copy the methylation pattern to the nascent DNA strand after replication. And the way it's able to do this is through its main cofactor, UHRF1, which has been shown to tether DNMT1 to chromatin via its own methyl DNA binding domain. Therefore, there's like a faithful copy of DNA methylation patterns after cell replication. People have generated DNMT1 knockout mice, and when you look at DNMT1 knockout ES cells, they actually proliferate normally. But like many ES cells that are defective in epigenetic modifiers, when you ask them to differentiate, they can't do that properly. And people have noted that in I'm so used to working on DNMT3L, but DNMT1 knockout ES cells, you see elevated mutation rates, retrotransposition, and actually reduced genomic stability as well. So DNMT1 knockout mice show abnormal growth, and they actually die mid-gestation. They're shown here, the knockout mice with a wild-type littermate. And DNMT1 heterozygotes and those mice carrying hypomorphic alleles have actually been analysed in a lot of different cancer models. And what they see is there's differing outcomes depending on the model that's been investigated. So depending on the cooperative genes, the cell type that's being mutated, also even the stage of disease progression, whether it's metastatic or not. So sometimes DNMT1 haploinsufficiency will actually accelerate disease, where in other cases it might actually slow it down. And this most likely depends on whether that particular tumour type is actually depending on genomic instability or tumour suppressor hypermethylation for its replicative ability. So DNMT3 family, these are the de novo methyltransferases. So they actually lay down the DNA methylation pattern. They're able to methylate both unmethylated and hemimethylated DNA. And the two family members, when you knock them out in mice, they both result in lethality, but at different stages. And this may be due to some compensation between the activity of the two proteins. So the DNMT3A knockout mice shown here actually appear normal at birth, but then they become runted and die at around 4 weeks of age. Whereas the DNMT3 knockout mice are embryonic lethal at mid-gestation, shown here. And the last family member, DNMT3L, as I mentioned, has no catalytic activity, but it is a regulator of methyltransferase activity. It's able to bind to and stimulate both DNMT3A and 3B. Knockout mice of DNMT3L have been generated, and these mice are actually viable and they live happily to old age, but both males and females are sterile, and that's because they have a failure to actually imprint parental imprinted loci properly. And in the male germ cells, you actually see a failure to methylate transposable elements. And so shown here is a wild-type testes section and a DNMT3L knockout testes section, and you can see due to the failure to methylate parental imprints and repetitive elements, you actually have a failure to generate mature sperm cells, shown by the absence of sperm here. So this brings me on to my first disease that I'd like to talk about today, which is actually when mutations occur in de novo methyltransferases. And this disease is called Immunodeficiency, Centromeric Instability and Facial Abnormalities Syndrome, or ICF. And this is an extremely rare autosomal recessive disease that actually affects about 1 in 1 million people. And the major symptom of this disease is immunodeficiency, with a reduced serum immunoglobulin level. And it appears that in people with this disorder, they actually have a failure to properly differentiate their B lymphocytes. And when you look in the peripheral blood, you see immature but not mature B lymphocytes. And due to this immunodeficiency, people with this disease often succumb to infections in early childhood. Other features of the disease are facial dysmorphisms and developmental delay. In about 60% of cases of ICF syndrome, they've found mutations in DNMT3B, so the de novo DNA methyltransferase. And they find that mutations generally target the catalytic domain, but they don't result in complete loss of enzymatic activity. And this is probably understandable given the embryonic lethality of DNMT3B deficiency in mice. So what they've found that in B lymphocytes, when you examine those, there is aberrant hypermethylation, shown here, of the satellite repeats that normally sit around the heterochromatic centromere region. And this is particularly evident on chromosomes 1, 16, and to a lesser extent, chromosome 9. And this results in hypermethylation, a loss of heterochromatin around the centromere, and therefore chromosome instability. And in these lymphocytes, you can see these multiradiate chromosomes shown here, where there's various bits of chromosome 1 and 16 sort of all stuck together. And it's thought that this chromosome instability, as well as dysregulation of genes, like DNMT3B target genes, actually results in a defect in B lymphopoiesis in these mice and humans. So that was an example of what happens when you have a defect in the DNA writing enzymes. What about when you have how exactly does DNA methylation result in gene silencing in the first place? Because, of course, there we saw a loss of silencing at the heterochromatic pericentric regions. So there's two real mechanisms that people think about when they think about how DNA methylation causes silencing. One is that it prevents transcription factors from accessing their recognition sites. But the far more prevalent way in which this acts is that there's actually the recruitment of methyl Cpg binding proteins. This in turn recruits histone-modifying enzymes and a whole slew of epigenetic modifiers that work together to mediate chromatin compaction and transcriptional silencing. And I think it's really important to point out here that while I'm mainly talking about DNA methylation doesn't act in isolation. It's one of a multitude of different epigenetic modifications that work together along with, like for example, most of the DNMTs can actually interact with histone deacetylases. They recognise different histone methylation marks. And so these components all act together in order to achieve chromatin compaction and transcriptional silencing. So what happens when you have a mutation in something that actually interprets the DNA methylation pattern? And that brings me on to the next disease, which is Rett Syndrome, or RTT. There was a previous speaker in this series, Joseph Getz, who actually talked about Rett Syndrome, and he gave a really great insight into the clinical side of the syndrome and also how mutations in MECP2 were found to be causative for this syndrome. So I'm going to concentrate more on how mouse models have been used to sort of elucidate what's going on in this disease. But I will give you just a bit of a refresher of the disease and the symptoms. So Rett syndrome is an X-linked neurological disorder affecting about 1 in 10,000 females. You've got to remember here, of course, that mammals are XX, and so they're actually mosaic for this mutation. So in some cells, they're going to inactivate the wild-type copy, and in other cells, they're going to inactivate the mutant copy. And usually this is a random event, but they've actually seen in some female patients that there's a skewing towards keeping the wild-type copy active and the mutant copy inactive in the brain. So in girls with Rett syndrome, they actually develop normally. They have normal prenatal and perinatal development. And it's then in early childhood, at about 6 to 18 months of age, that they start to digress. And they see loss of acquired linguistic, social, and motor skills. Then they start to develop autistic features, stereotypies, and seizures. One of the really interesting parts of this disease, though, is that they don't see neuronal death in these patients. They actually just see abnormal neuronal morphology rather than apoptosis of those cells. There are very few affected male cases. It's very rare, as you can imagine, because males are hemizygous for the X, and so all of their brain cells would be expressing the mutant copy. And the males seem to fall into these two categories, those with very severe symptoms that don't survive infancy, and those that can actually survive for decades with Rett-like symptoms. And this correlates very, very strongly with the type of mutation that they have. So in the first group, they often have very early truncations of the protein, so only the N-terminal region is expressed, whereas in the second type, they have much later truncations, so that you're only losing small amounts of the C-terminal region. And as I mentioned before, Rett syndrome is caused by mutations in this methyl Cpg binding protein, MECP2. It's a ubiquitously expressed protein, but it's actually very, very highly expressed in postmitotic neurons. And it has its methyl Cpg binding domain, but it also interacts with a bunch of different chromatin modifying enzymes, and it's known to also be involved in chromatin compaction. So there are several mouse models of Rett syndrome. There's complete knockouts, conditional knockouts, which are only in postmitotic neurons, and they've also introduced specific human mutations into the endogenous MECP2 mouse locus. And you do actually see Rett-like symptoms in female knockout and male hemizygous mice. And these include limb clasping, breathing difficulties, and they also have reduced brain size and body weight. And similar to what we see in the human disease, there's actually a delay in the development of symptoms. So symptoms arise at about 5 to 6 weeks of age, and then you have mortality at about 2 to 3 months. They also looked at MECP2 heterozygous females, so these are most similar to the human disease, and actually have a really similar delay before, as in humans, despite different ages of humans and mice, obviously, but they actually get symptoms at about 6 months, and they have normal lifespan as well. So the fact that symptoms are quite dramatically delayed in some cases suggested to researchers that it's not the development of the brain that's an issue, but the stability of brain function. And so they thought perhaps there are ways of treating this so that the symptoms are actually reversible. And this is something that Adrian Bird's group were able to do with MECP knockout mice. So in these mice, we have here a 12-week-old MECP2 hemizygous male, and you can see that it's not moving around very much, and it has really laboured breathing, and you'll see later on when it gets picked up it does this sort of limb clasping behaviour. But these mice have been engineered so that with tamoxifen treatment you can actually reintroduce MECP2 expression, and that's an example of the mouse here. So you can see that it was running around quite happily and it doesn't have that laboured breathing anymore. And in addition to that, they're actually able to extend the lifespan of mice that have been tamoxifen treated and therefore reintroduced MECP2 expression into their brains. And I'm sorry that that's a terrible picture to end on there. They also looked at this in the MECP2 heterozygote mice, and the female mice with late onset symptoms, and saw a similar reversal, or quite robust reversal of some of the symptoms. So this is actually very promising when we consider whether or not there's any therapeutic options in humans. And whereas we can't engineer humans so that we can re-express MECP2 with tamoxifen treatment, we can look at some of the downstream misregulated genes in MECP2 knockout neurons and see if we can perhaps reintroduce some of these factors. And two of the factors that I found people were looking at were brain-derived neurotrophic factor. And this is actually underexpressed in MECP2 deficient neurons, and there's a small molecule here, CX546, that when you treat mice with this, you see a robust upregulation of this factor, and it actually improves the respiratory symptoms in these mice. And another factor that people are looking at is insulin-like growth factor 1, and this is really important in neuronal maturation and synapse plasticity. And I recently saw that there's a clinical trial treating girls with Rett Syndrome with recombinant IGF-1, and that just finished at the end of January this year, so hopefully there'll be some interesting results coming out of that. So Rett Syndrome is a great example of what happens when you can't interpret the DNA methylation code properly. So next I kind of want to change tack a little bit and talk more about when in development you actually see these methylation patterns being established. And so that brings me to this schematic here. And so during reprogramming there seems to be two really essential periods where you get almost complete erasure of the DNA methylation patterns, and then it gets re-established in a tissue or cell type specific manner. And that's in the pre-implantation zygote, and then again in the primordial germ cells. Because generally you find that the bulk genomic CpGs, their methylation status is actually quite stable across developmental time once they've been established in the embryo. And even though there are more and more reports of quite dynamic changes in DNA methylation at certain promoters, this seems to be more specific to defined cellular processes within a specific cell type. So for example, CD4 T cells deciding to become Th1 or Th2 responders, that might involve some rapid dynamic DNA methylation changes. But generally, the bulk DNA methylation remains consistent. And that's why these two periods where there's complete erasure of DNA methylation are actually really interesting and important. So we should probably start at the very beginning, which is with the oocyte and the sperm. And these two cell types are actually quite highly methylated compared to somatic cell types. And so this table here is a summary of some amazing work looking at the genome-wide DNA methylation studies on germ cells and early embryos. So I'm showing this a few times, so I'll just take you through it. We've got the scale of CpG methylation here, where black is really highly methylated and the kind of off greeny grey colour is low methylation. We have the different cell types that they've looked at here, so sperm, oocytes, early embryo, and these are different stages of primordial germ cell development. And then we have some of the DNA elements that people look at when they talk about DNA methylation. So global genome methylation, the paternal and maternal imprinted loci, and then also the repetitive elements. So particularly when we're looking at paternal and maternal methylation, they've actually been able to look specifically on either the paternal genome or the maternal genome for each of these elements. So if you take sperm, for example, it doesn't have a maternal genome, it only has a paternal one, and you can see the paternally methylated ICRs are actually paternally methylated, and similar in the oocyte, which doesn't have a paternal genome, the maternally methylated ICRs are maternally methylated. So once egg and sperm meet and they become a pronuclear zygote, they have to undergo massive DNA methylation changes. In fact, once the sperm actually enters the egg, it has to undergo massive changes. There's the replacement of protamines with histones and decompaction of that really tightly compacted sperm chromatin. But what's interesting is the way that the different genomes are treated are quite differently during this period of demethylation. So the maternal genome is actually passively demethylated, while the paternal genome is actively demethylated. So what I mean by passive demethylation of the maternal genome is this is achieved by actually excluding the maintenance methyltransferase, DNMT1, from the nucleus in the zygote. So as you can see here, DNMT1 is shown by that nice white staining here, and it's not in the nucleus, it's actually out in the cytoplasm. And this is consistent at the 2 and 4 cell stage. So by removing DNMT1, not letting it do its job in the nucleus, you actually get this passive demethylation of the maternal genome. And then once you hit the 8 cell stage, it's located back in the nucleus again. And this is shown by schematic again here, where red illustrates the DNMT1 localization. In contrast, there's active demethylation of the paternal genome. And so here, this is a mouse pronuclear stage zygote, and this is the time post-fertilization. And what you can see, we've got the male and female genomes here. And at about 8 hours post-fertilization, I should point out that DNA is marked here in the blue, whereas green is 5-methylcytosine. So you can see at this stage, both the male and female pronuclei are actually methylated. But then when you move into the 8 cell stage, you can see there's been this rapid loss of DNA methylation on the paternal genome, but not the maternal genome, and complete decondensation of the DNA here. And these are cells that have actually been treated so that they cannot divide. And you can see this is still true, which means that this is a replication-independent process, this active demethylation. And it's been quite recently that we've actually had some insight into how DNA demethylation, active DNA demethylation, occurs. And that seems to be via this intermediate called hydroxymethylation. And this is mediated by TEP proteins, which you've probably heard of. And so again, a picture of a mouse zygote here, you can see the maternal nucleus here is marked red for 5-methylcytosine, whereas the paternal is marked green for 5-hydroxymethylcytosine. So for many years, and certainly while I was doing my PhD, the way in which active demethylation occurred was pretty much a complete mystery, and it has quite a colourful history of people trying to say whether or not, oh, maybe the DNA methyltransferases actually demethylate as well, and perhaps there are some DNA Cpg binding proteins that actually demethylate. But it's only been recently that we've had a bit more mechanistic insight into how this happens. And it seems to be mediated by this TET family of oxidases. And my knowledge of this is still quite basic, so I'll keep it simple. But what seems to happen is that methylated cytosines are then oxidized and converted into a form that allows them to go through the base excision repair pathway, and therefore they're replaced with unmethylated cytosine. And so this is a way of replacing methyl with unmethylated cytosine in an active manner. So during this massive period of demethylation, both passive and active in the zygote, there are certain sequences that are actually protected from methylation, and these include the parental imprints and the repetitive elements. So you can see here, looking at the parental imprints, you can see that on the paternal genome, the paternally methylated imprints actually stay methylated in the blastocyst and epiblast, and similar, the maternally imprinted and methylated sequences stay methylated on the maternal genome. That was a mouthful. And similarly, you see that you don't really ever see a large drop in methylation on the repetitive elements. And they've recently found that it is due to these maternal factors that are present in the oocyte that actually protect these specific loci from being demethylated during that big demethylation phase. And I've just given 2 examples here. One of them is stellar, and it's actually able to bind to parental imprinted loci by recognising a particular histone modification. That prevents TET3 from binding and therefore prevents the conversion of 5MC to 5HMC. And another one of these factors is TRIM28 or CAP1, and this actually targets DNMTs specifically to parental imprinting control regions, and therefore makes sure that the methylation on these areas is maintained. And so finally, once you develop the embryo in the various different tissue types, there's a second period where you see this global demethylation, and this is in the primordial germ cells. And what has to happen here is you've got these, all of these sort of methylation patterns have been established during this period of development, and then you need to wipe them completely again and re-establish germ cell-specific pattern, including imprints that based on the parent of origin, so either male or female imprints. And so here I'm just showing a schematic of the reprogramming that occurs during gametogenesis. So this represents the amount of DNA methylations going on, and so you see this loss of methylation quite rapidly, and then you see an increase of methylation again in either a sperm or oocyte-specific manner. And this happens at different times during development because, of course, these are quite different developmental processes, spermatogenesis and oogenesis. And I should again re-emphasize that it's only been recently that they've been able to, you can imagine there's very few of these sort of cells, so it's only been recently that we've been able to gain some insight into the global DNA methylation patterns. And at this point, we really only have information on DNA methylation and not necessarily on histone modifications, because we haven't been able to do sort of genome-wide chip analyses in these cell types yet. So what do we know about this demethylation process? We know that, spookily similar to what happens in zygotes, it happens in both an active and passive manner. So there seems to be a first wave of methylation in the migrating primordial germ cells, and in mice this is at about E8.5. And this is a global demethylation, and again, this seems to be mediated by, in this case, relegating DNMT1's cofactor out of the nucleus and into the cytoplasm, so it can't do its job in maintaining DNA methylation patterns. And we also see a downregulation of the de novo methyltransferases, DNMT3A, 3B, and 3L. There then appears to be a second wave of demethylation, and this happens as the primordial germ cells migrate once they've actually entered the genetic ridge, the genital ridge, sorry. And this second wave seems to occur at around E11.5 in mice, and this is where the areas of the genome that are usually actually really tightly methylated, so really strongly methylated and repressed, such as the inactive X, germline-specific Cpg Islands, and also the paternal imprints, it's there at this stage that these really strong methylation marks are actually wiped. And there is a thought that this might be involving 5-hydroxymethylcytosine. And then again, we need to re-establish these imprints by de novo methylation, and this again requires the de novo methyltransferases. So I've spoken a lot about genomic imprinting, and so now I'm just going to kind of go back and talk about genomic imprints a little more, parental imprints. And I wanted to first share this kind of fascinating story that I came across. And this was in 1638, there is evidence of a woman called Mrs. Anne Hutchinson, and I think this was like her 14th pregnancy, but she actually gave birth to what was described as a handful of transparent grapes. And the Reverend John Cotton, who was attending at the time, commented on this in a sermon, saying that it seemed to be several lumps of a man's seed without any alteration or mixture of anything from a woman, which is a fairly horrible description, but was actually quite accidentally insightful, because what he was most likely describing was a complete hydatidiform mole, shown here. And so this is when the conceptus has 2 sets of parental chromosomes and no contribution from the maternal genome, and is formed entirely of trophoblast. So it was quite early on that people realised that there was a functional non-equivalence of the paternal and maternal genomes. So you cannot make an embryo with either 2 female or 2 male pronuclei. And these experiments were done in the 1980s by both the Salter and Serrani labs. And they showed, whereas course, you can get a normal conceptus from a male and female pronuclei, but if you put in 2 females, you fail to develop the extra embryonic lineages, and 2 males, you fail to develop the embryonic lineages. There is something fundamentally different between the male and female genomes. And this is the genomic imprints. And so when we talk about genomic imprints, we talk about the monoallelic expression based on a parent of origin of the allele. So this is a very overly simplified version here, but what I'm showing is a maternal genome here and the paternal genome here. And we have this one gene, in which case there's differential methylation between them, and I'll explain it's not quite as simple as methylated means silenced and unmethylated means expressed. But what you can see here is that we have expression of the maternal allele where the paternal allele is completely silenced. And early evidence for genomic imprinting was due to the fact that some deletions were actually only pathogenic when they were inherited maternally, but not when transmitted through the male germline. So you can imagine if there's a mutation here and you get that from your mother, of course it's going to be expressed and there's no alternate copy to sort of protect you from that mutation. Whereas if the mutation's here and the gene is silenced, it doesn't actually have an effect. And the way people first started to map genomic imprints is that they generated what they call uniparental disomies, or in some cases, uniparental duplication deficiencies. So this is when perhaps you have 2 paternal copies of chromosome 7 and no maternal copies, or in the duplication deficiency, certain sections of chromosome 7 that both male, no female contribution. And they noted in some of these embryos that there were abnormalities in growth and viability. Today in mouse, we know that there are around 100 imprinted genes, and many of these are located in clusters, shown in this diagram here. And it's actually quite a complicated process because they often see that there is tissue-specific imprinting, so there are some genes that are only imprinted in the placenta or the brain. And then you often see cell-type-specific absence of imprinting, and this seems to be particularly evident in the brain, where you sometimes see biallelic expression of genes such as DLK1, which are normally monoallelic expressed in all other cell types. So genomic imprinting is controlled by the imprint control regions, and this is associated with DNA methylation at this region. And there are 2 main mechanisms that have been elucidated of how genomic imprints work, and these are long non-coding RNAs and insulator or enhancer blocking. And so as I noted, it's not as simple as DNA methylated means silence like it is in Cpg Islands. And so here's an example of a maternally methylated region, the KCNQ1 region. And so the ICRs are actually shown by these little circles here, where the dark colour means methylated, and if it's white, it means unmethylated. You've got the maternal in a nice red and the paternal in blue. And you can see paternally expressed genes are shown in blue, while maternally expressed genes are shown in red. So in some cases in these loci, you also have biallelically expressed genes in black, and those genes that are silenced are in grey. So the way that this locus actually works is you have maternal methylation of the maternal allele, so this occurs in the developing oocyte, and this is actually in the promoter region of a long non-coding RNA, KCNQ1OT1. And so because it's unmethylated on the paternal allele, you get expression of this long non-coding RNA. It recruits a bunch of histone-modifying enzymes, including PRC2, and that actually silences this region in cis. And then because you don't have methylation on the maternal side, you don't get expression of this long non-coding RNA, and so expression of a number of these genes is exclusively on the maternal side. And I'd particularly like to point out this CDKN1C gene, and this is maternally expressed cell cycle regulator involved in growth restriction. Now the second example I'd like to give is of a paternally methylated locus, and this is the IGF2H9T locus. You can see in this case the ICR is actually located in an intergenic region rather than at a promoter, and it's methylated on the paternal side. And so the lack of methylation on the maternal side allows this protein called CTCF to bind. And the binding of this protein actually blocks the use of these enhancers with these genes. And so instead, you have expression of this H19 long non-coding RNA. Whereas on the paternal allele, there's looping that allows these enhancers to cause the expression of insulin-2 and IGF-2. And I again want to point out this gene, IGF-2, which is paternally expressed, and it's actually involved in growth promotion. So the example of an imprinting disorder, and this is when imprinting goes awry, that I'd like to give is Beckwith-Wiedemann syndrome. And this is a fetal and postnatal overgrowth, has fetal and postnatal overgrowth symptoms, and this is associated with an increased risk of childhood tumours, particularly of the liver, kidney and pancreas. So this results in abnormalities in these two loci I just mentioned that are actually linked and are on 11q, chromosome 11q, as shown here. And so this abnormality arises when you have maternal alleles that behave like a paternal allele instead. So if the maternal allele is like a paternal allele, you're losing the expression of this growth-restrictive CDKN1C, and you're gaining overexpression of this IGF2, which is growth-promoting, therefore leading to an overgrowth syndrome. And this can occur due to deletions or rearrangements in the maternal chromosome so that you don't get proper imprints. It can also be caused by uniparental disomy, in this case 2 copies, paternal copies of chromosome 11, and also due to just abnormal methylation, so a failure to methylate the ICRs properly, which will cause, although this is quite rare. So lastly, I'd like to talk about repeats and some of the diseases induced related to repeat-induced instability. And so here again, I'm showing the repeat elements, lines and IAPs. So lines do actually go through periods of hypomethylation, but they don't seem to be expressed during development, whereas IAPs, which are developmentally or evolutionarily much younger elements and therefore more likely to be able to sort of jump around and cause havoc, maintain really, really high levels of methylation through development. And of course, the reason we don't want transposable elements to be unmethylated and expressed is because they can jump into coding genes, causing mutations. There's also the possibility of using the strong promoters associated with these repetitive elements cryptically so that they might overexpress neighbouring genes or aberrantly express neighbouring genes in the wrong cell type. And it's also to prevent a legitimate recombination between repeats. So the last 2 diseases that I'd like to talk about quickly are involved in repeat instability. And the first one of these is Fragile X syndrome, which you've probably heard of. And it's a common cause of mental retardation occurring in about 1 in 4,000 males. And this is an X-linked disorder that maps to the FRM1 gene. And this is really highly expressed in neurons. And it's an RNA binding protein that seems to be involved in signaling through the synapse. In a normal person, you would expect to have this is the MFR1 gene shown here. You have a number of these CGG trinucleotide repeats. And in a normal person, this is usually below 50 repeats. And so the area is not methylated and you get expression of the gene. But what happens in Fragile X syndrome is that you have this massive expansion of this repeat. And so you end up with over 200 copies. This then attracts methylation enzymes. You get methylation of the repeat and you actually silence the FRM1 gene in neurons, therefore causing the disease. And then the last disease I'd like to talk about is FSHD, which is a muscular dystrophy. And this actually, again, has to do with repeats, but as opposed to being an expansion of repeats, it's a contraction of repeats that causes the disease. So this is an autosomal dominant inherited form of muscular dystrophy, and it affects the skeletal muscles of the upper body with a prevalence of about 1 in 20,000. Now, it's associated with these macrosatellite D4Z4 repeats shown here in the blue triangles. And these are units of about 3.3 kb, and these are repeated in the telomeric region of chromosome 4. And again, in normal people, you tend to have somewhere between 11 and 150 copies of this repeat, and this is epigenetically silenced, as shown here by the black circles that represent Cpg methylation. Each one of these D4Z4 repeats actually contains a copy of a gene called DUX4, or double homeobox4. And normally the expression of this gene is restricted to germ cells and pluripotent cells. And now this syndrome actually becomes more difficult because you only get it on a certain genetic background, and that's if you have this permissive variant. And there's 2 different variants, 4QA and 4QB. And 4QA actually contains this polyadenylation sequence. And what this does is if DUX4 is to be expressed, it actually stabilises the mRNA of DUX4 and allows for it to be expressed, stably expressed aberrantly in skeletal muscle. So what happens in FSHD is you get contraction of the D4Z4 repeats, And this, in combination with a permissive variant, actually causes this disease. So shown here, you have hypomethylation, inefficient epigenetic repression, and therefore a relaxed chromatin state. And when this is in combination with permissive allele, you get variegated expression of DUX4 in the skeletal muscle. And this is really interesting because instead of seeing, say, a low level of DUX4 in every single muscle cell, what you see is a few muscle cells express really, really high levels of DUX4. So it's actually a variegated expression. And with the expression of DUX4, you have increased expression of other of its downstream targets, which are other pluripotent and germ cell-specific genes, and this is thought to cause apoptosis of the skeletal muscle cells and kind of describes the progressive nature of the disease. But of course, it's always more complicated, and there is also this version of the disease called FSHD2, and this is kind of symptomatically indistinguishable from FSHD1, although of course there is some variation in the phenotype in these people. It also has an autosomal dominant inheritance pattern. And what you see in this case is you get hypermethylation, chromatin relaxation, and DUX4 mRNA expression, but it's independent of the size of the G4Z4 repeat. And there's recently been a great paper that has actually identified the mutation associated with FSHD2. And using whole exome sequencing, they identified heterozygous mutations in a protein called SMOOCH-D1. Now, this is an epigenetic repressor that's involved in mice in X inactivation and also repeat-induced transgene silencing. So a really good candidate for this disease. And what they see in this case is when you have a heterozygous SMOOCH mutation, you have reduced levels of SMOOCH, and so this leads to relaxed chromatin at D4Z4 and expression of the mRNA, even in the absence of the contraction of the repeat. So that's the last of the diseases I wanted to talk about. So now, just to summarise, I hope I've shown you the importance of DNA methylation during development and these really interesting periods of complete erasure and re-establishment of DNA methylation patterns. But I just want to remind you that DNA methylation is one of many epigenetic modifications that act in concert to regulate cell identity and function. And as we learn more and have these sort of genome-wide studies, we'll be able to sort of place all of these things in order of importance. The diseases I've talked about today are really the classic chromatin diseases, but it's becoming more and more evident that a lot of these epigenetic modifiers are actually playing really important roles in other diseases, in particular cancer. And Catherine in her seminar alluded to the fact that DNMT3A and TET2 are actually driving mutations in AML. Also in cancers, you do see this aberrant DNA methylation and loss of imprinting. So I think in both ways, sort of like studying the reprogramming and normal behaviour of these modifiers will help inform how we design treatments for cancer. And when we're actually designing treatments for cancer, it's important to remember that if we disrupt these epigenetic modifiers, that they are most likely going to have an effect on other cells in the body. And we should need to be particularly mindful of germ cells. And finally, I think understanding these periods of natural reprogramming and the immense demethylation and remethylation in germ cells in the early embryo will hopefully inform us into better ways to generate induced pluripotent stem cells, which of course are a promising therapeutic for the future. And finally, I'd just like to thank members of the lab and friends who have listened to me talk about my talk in the last few weeks. So thank you.
Thank you for a great seminar, Sarah, and lots of molecular mechanisms, which are always really interesting. So student questions first.
So in an experiment, the Cpg is often summarised as being either methylated or unmethylated, but when you look at bisulfite sequencing data, you often see it's perhaps 10-20% methylated or 80-90% methylated. How do you interpret those? Is it technical artefact, or do you think there's a subpopulation of cells that have a different methylation state?
It could be any of those things. It could often be heterogeneous cell population, therefore you're seeing some unmethylated, methylated. But I think even when you have really high cell purity, you probably actually do see some differences in methylation. And that, in a lot of cases in these diseases, it might actually explain why you do see such like phenotypic variation in the symptoms that come out, because, you know, at those various levels of methylation might just tip you over from one side to another. So I think in a lot of cases, it's probably actually genuine variation.
Just a background question, because this isn't my area and maybe I heard wrong at the start, but you said that DNA methylation is differentially disrupted in cancer, so you get hypermethylation globally and hypomethylation, which is
Opposite way.
That's what I was going to ask.
It was written on the slide, but yeah, I probably just stumbled over my words. So generally you see hypomethylation globally, and this is thought to sort of make the genome less stable in those cells, and then you have specific hypermethylation and silencing of tumour repressor genes.
Any last questions?
Sorry, could you say something a little bit more about how the DNA methylation, you seem to imply that the more CpG marks there are, sequences there are in an area, the more likely it is to be methylated or demethylated, or?
It seems to be really context dependent from what I understand. So CpGs and repetitive elements are more likely to be methylated. But you can have really dense Cpg Islands in like upstream of a housekeeping gene, and they're more likely to be hypomethylated, so undermethylated. And I think a lot of the time it's very chicken and egg on how this happens, but it might actually be the fact that that is transcribed means it won't be methylated, versus the fact that this bit, is methylated means it won't be transcribed. So there's kind of a balance between those two things there. So I think it really is context dependent, and there are a lot of different factors that say specifically make sure that repetitive elements are hypermethylated, whereas they may not function in Cpg Islands.
Okay, so let's just thank Sarah for a fantastic